pleasure to be here with you all, and an honor to be a part of this program. I really enjoyed the work that we're doing together on virtual cell models with John and so on. And I'm glad that you'll get a chance to hear about the work that's happening shortly. I'm going to tell you about efficient AI-based experimental design, which is based on collaborations with a number of wonderful collaborators, both on the AI side and on the experimentation side. So in drug discovery and also in materials discovery, smart experimental design is a critical part of accelerating the process. We view this as an iterative loop where I perform experiments, I observe what happens, and then traditionally a human, and increasingly an AI system, interprets the data and then makes decisions about what experiments to run next. Physical experiments are time-consuming and laborious to perform, and so by being more intelligent about data interpretation and choosing which experiments to perform, we can reduce the time required to discover a new drug, discover a new material. Historically, much of the progress from an AI perspective has been made through the use of black box optimization methods. These methods view the system that I'm trying to optimize. In the case of drug discovery, I'm trying to maximize some therapeutic, some health outcome, while subject to constraints on safety. And we view the objective function and any constraints as being a black box. We don't know anything about this system, and the only way that we can learn about it is from the history of experiments that we've performed. And if you think about human health, some particular health outcome from a disease, when we measure it, the measurement is noisy. The function that maps, the design of my monoclonal antibody or my small molecule onto some health outcome, that function doesn't have any particular, nice shape that would allow me to optimize it efficiently. So I can't use classical convex optimization. One of the classes of AI methods that has seen the most success over the years is maybe I'm just going to do this. Bayesian optimization is one of the most widely used and successful AI-based methods for optimizing these black box functions. These methods have two steps. The first is a data interpretation step. We take the experimental data that we've observed and we fit some kind of predictive model to this data. Here what I'm showing you is an experimental setup where I have an input parameter, x, that ranges between 0 and 1. Maybe in a drug formulation design setting, you might think about this as the concentration of some particular ingredient that's make pictures easy for you to see, I'm only focusing on one input dimension in this optimization function. And on the y-axis, I have some figure of merit, some objective function. You can think about it as maybe something that I can measure in the lab that relates to the performance of this drug formulation. Maybe it's a solubility measurement, or a membrane permeability measurement, or maybe some biochemical activity. In this example, I want it to be as large as possible. So I've observed this outcome at 4 different input points shown in green circles, and I fitted a machine learning model. Here, in this case, is what's called Gaussian process regression. If you're not familiar with Gaussian process regression, just think of it as a machine learning technique that's able to model continuous functions and that provides both an estimate and quantifies the uncertainty associated with that estimate. So in this image, I'm showing the actual function, which is a dashed green line, and then I'm showing the estimate from this machine learning model in the solid blue line, and some estimate of the uncertainty here in the shaded blue region. Okay, so that's the first component of Bayesian optimization. It's some statistical, some machine learning model, and critically, it quantifies uncertainty using Bayesian statistics. Then, using this model for the data, which comes in the form of a probability distribution, which we call a posterior probability distribution because it comes after observing the data, we decide what experiment to perform. And this decision is based on an analysis of the value that the experiment provides towards my goal of, in this case, optimizing the objective function. Okay? So, and I'll describe in a little more detail in a bit how this function is computed, but you can see that it tends to be large at places where the blue line is large, so the estimate is large, and also at places where the uncertainty is large. So this is the classic, you may have heard in reinforcement learning, of the exploration versus exploitation trade-off, which is telling us that if we want to optimize something, we want to measure at places that we think are good, and also at places where we don't know what will happen. This acquisition function is also 0 at places where we've already measured, because in this particular example, the amount of noise that I have in my measurements is relatively small. We then use an optimization method to find the experiment to perform that maximizes this acquisition function. That's easy to do because this acquisition function is fast to compute, unlike my laboratory experiments, which take weeks. I can compute this acquisition function in less than a millisecond. I find this particular concentration of my ingredient, and that would be the experiment that I run next. I get that data, right? So I get a new green point. I update my Bayesian posterior probability distribution, recompute it, and optimize my acquisition function, find a new experiment to perform, and repeat. Okay? So if I've done a good job of setting up my machine learning model for quantifying uncertainty and making estimates, and done a good job of optimizing the acquisition function, then in this example, after a relatively small number of experiments, I found a solution to my optimization problem here that is close to the global optimum. These methods work in an integrated way with large language models. You've seen in recent years a large number of black box optimization methods that are leveraging these ideas to accelerate performance in iterative optimization in drug design and materials design applications, also in hyperparameter optimization for machine learning, and also a variety of other applications. You have some work which is thinking about using an LLM directly as both the data interpretation and the decider about what experiments to perform. You have a collection of methods in which Bayesian optimization and the kind of classical Bayesian optimization that's using often these Gaussian process-based machine learning methods are working together with LLMs, where LLMs will propose experiments to perform in high-dimensional spaces, and then Bayesian optimization will then choose which experiments are most informative. My group has a recent work that we just presented at a ICML workshop last week. And then you have also even tighter integration between large language models and Bayesian optimization approaches in which you're using the LLM inside of the predictive methods. So I've been applying these methods in the context of drug and materials discovery for several years. And in many ways, the methods are wonderful and do amazing things. But there are gaps. And I want to illustrate one of what I view as most important gaps using an example of a drug. So this is Paxlovid. It's a drug for COVID-19 and the virus that causes COVID-19, SARS-CoV-2. It's given to high-risk, non-hospitalized adults early in the course of their infection before they get too sick, that have been infected by SARS-CoV-2 and are symptomatic. And this drug actually has two active ingredients. So nirmatrelvir, which slows the ability of the virus to correctly reproduce itself, and ritonavir, which was a molecule that's been used in other drugs, in particular antivirals. It's used in some HIV drugs. And it makes nirmatrelvir last longer in the body. It's very effective. So this is some data from the clinical trial, and one of the clinical trials. And it reduces hospitalization and death by almost 90% compared to placebo. So if you think about this as a black box optimization problem, you think, well, AI is amazing. How can I apply these methods to solve this important? Like, if Pfizer makes this drug, if I were Pfizer and I hadn't yet produced this drug and it were early in the pandemic, how would I have used the power of AI to more quickly develop this drug? So the first thing you would do is you'd write down, what is my objective function? Well, it's effectiveness, right? So maybe let's make that a little bit more precise. I'm going to try to reduce all-cause hospitalization and death over some period which is relevant for the progression of COVID. And then there's also a constraint on safety, right? So the number of adverse events of various types should be rare in the clinical trial population. So it's classical optimization problem. I optimize this thing subject to a constraint on this thing. Perfect. The problem is that evaluating the objective function and the constraint, when it's posed in this way, requires running a clinical trial, which is very expensive and takes a long time, right? So if I tried to literally run the loop, every time I got to the point where I wanted to perform an objective function evaluation on a particular proposed drug, and keep in mind here the drug is, Paxlovid, right, contains two active ingredients, and then any drug is formulated so that you can either take it orally, or it can be injected, or it can put it in your bloodstream or inject it under your skin, right? So there's a variety of other ingredients that allow those active ingredients to and be stored during transportation. So the design problem requires you to choose all of those things. So it's a high-dimensional search space, and here we're talking about one evaluation takes years and quite expensive. So that's not going to be easy. If you work in this area of black box optimization, maybe you've heard about multi-fidelity optimization. So this is an approach where I have a very time-consuming and expensive to evaluate objective function and constraints, and I have a less expensive to evaluate, we call it a low-fidelity approximation, right? And this is often, and then the idea is that you do lots of experiments on the low-fidelity system, the low-fidelity observation method, and then translate what you learn via some multitask learning method over to your original problem. We do this in drug discovery with animal models, right? This is one of the most widely used animal models, right? Mice and rats, where the physiology of these animals is similar to our own physiology. It's not exactly the same, but close. So we can observe, health outcomes, and then translate that over to human. But it's far from the whole answer because, all of these reasons, right? Life is precious. Running these experiments is also expensive. It's less expensive than running a human clinical trial. It's also slow. It's not as slow as running a human clinical trial, but it is slow. And, the outcomes are noisy. There's an opportunity to do much better, which is, if you only look at efficacy and safety when you run these animal experiments, you're ignoring a wealth of information which is causing you to take longer than necessary to do drug discovery. So if you think about how drugs are actually discovered, we bring to bear a wealth of knowledge about biochemistry and biology. So in particular, right, let's think about SARS-CoV-2 and what people at Pfizer were probably thinking about when they developed Paxlovid. So there is this enzyme, which is a protease, which chops up proteins. It's called 3CL protease. And this is an enzyme which is important to SARS-CoV-2 and allows it to reproduce. Okay? So it's an enzyme that it needs to copy itself correctly. If the virus doesn't have 3CL protease, then its copies are going to be malformed and it's not going to be able to successfully reproduce itself. So the core idea in this drug, Paxlovid, is we're going to create a molecule, later named Controllable, that binds to 3CL protease and prevents that complex, the bound complex, from allowing SARS-CoV-2 to do the kind of copying that it needs, right? So if you increase this drug, then in the bloodstream in areas, or in the proximity of where SARS-CoV-2 is doing its reproduction, then you're going to reduce the amount of SARS-CoV-2 in the body, right? And when Pfizer was developing this drug, presumably one of the things that they did was to modify this small molecule, the structure of this small molecule, in order to improve the strength of binding, so as to more successfully prevent reproduction. One of the problems with this drug and with many drugs is that the active pharmaceutical ingredient doesn't last in the body as long as we would like, right? So you start to expand out this. I just showed you a simple diagram which was thinking about inputs and outputs, about the relationship between interventions that we perform on the human and things that happen inside the human and health outcomes that result, right? So this molecular structure, yes, it determines binding to 3CL protease, but it also determines the rate at which your body takes this molecule and chops it up and excretes it, right? So if metabolism is faster of this molecule, then you're going to have less availability of the molecule in your body, and it's going to have a smaller ability to bind to 3CL protease and prevent your virus from spreading, right? So this molecular structure determines it, and also at the top of the slide, the dosing, the frequency with which you can give somebody this nirmatrelvir drug is going to influence how much of it is around. Okay? So Pfizer was thinking, hey, we also want, we want to figure out a way to make this bioavailable enough that we're going to prevent, going to cure people. So there's this trick that people who work on drugs for viruses know, which is that we can impact the rate at which this metabolism happens through another intervention on the body. So there's this enzyme that we all have called cytochrome, I think, and so this affects the rate of metabolism. And if we give someone another small molecule called ritonavir, we can affect this enzyme so as to slow the rate of metabolism of the nirmatrelvir. Okay, so inside the mind of the people developing this drug, they're thinking about this diagram that encodes input-output relationships, right? They're trying to choose the molecular structure of nirmatrelvir. They're trying to choose the dosing of nirmatrelvir. They're trying to choose the dosing of ritonavir, and the formulation of all of this stuff in terms of other ingredients, so as to maximize bioavailability of this active ingredient. And in order to do that, yes, metabolism is important, but also the solubility of the drug in your bloodstream, and the permeability of cell membranes to this drug, right? All of which are affected by the molecular structure. So they're trying to design this complex system of molecules and formulation to achieve this goal. What can they measure, right? Well, they can do experiments on the bench or in a biosafety cabinet in 96-well plates or with a robot, where they modify the molecular structure and they observe how that binds to 3CL protease. They can also do in silico experiments where they take protein structure prediction or generative protein structure prediction and use that information to guide an understanding of how molecular structure impacts binding. They can also measure in typically a separate lab experiment how metabolism impact, how the rate at which this gets metabolized in the presence of this enzyme and ritonavir, right? They can do experiments where they measure solubility of nirmatrelvir, also in a lab, and also permeability of cell membranes, important cell membranes, to nirmatrelvir. Then they can measure all of this complex biology approximately in a mouse, right? So that's an animal model. And they can also, they can measure this, right? So they can do an assay where they look at the amount of nirmatrelvir in a mouse, right? So you give the mouse like the different molecules in the formulation, and then you draw blood and you measure, how much nirmatrelvir is present. You can measure how much SARS-CoV-2 3CL protease is present in a bound and unbound state. And then you can also measure the concentration of virus inside of the mouse. And you can also look at, of course, mouse health outcomes. Right, so humans, when they design drugs, are thinking about all of this. So if we want an algorithm to be able to compete with a human and outperform a human so that drug development can be faster, presumably our algorithms need to do this too. And black box optimization methods don't, right? So it's like, trying to win in a race where your competitor is driving a car and you're running, you're just not going to win, right? So we need to design algorithms that know these relationships between these things. So probably you've heard of multi-objective optimization, right? Could you use multi-objective optimization to provide these capabilities, right? So you basically like, you treat subsets of the network as separate multi-objective optimization problems, right? So maybe you're gonna have a problem where you're optimizing the molecular structure of nontrelvir, and you're caring about permeability, binding to 3CL protease, solubility, and then some measure of the rate of metabolism, right? So those are like 4 numbers, and then you're searching for the molecular structure that explores the Pareto frontier of those 4 numbers. And then you would separately, look at ritonavir dosing and try to optimize ritonavir dosing in order to optimize this. So there are, and also nontrelvir formulation and nontrelvir dosing. The problem is, and this is I think the most widely used approach within AI for drug discovery, and it's semi-effective. The problem is, first of all, how do you know what parts of the Pareto frontier to focus on, right? So humans who have expertise in this area, they have an understanding of how soluble does it need to be, but what is the trade-off between binding the 3CL protease and rate of metabolism, right? So that's specific to this particular system, and it's not easy for somebody to know how to make that trade-off. It's also hard to know which subproblem to focus on when, right? So these are all different lab experiments. Which lab experiments should I do? How do I know when I've done a good enough job with the molecular structure and I can jump over to dosing? Also borrowing information between overlapping subproblems. When I think about ritonavir dosing, right, the right answer for that depends on the molecular structure of nontrelvir, right? And when I think about nontrelvir dosing, that also depends on this molecular structure. But what I need from the molecular structure depends on the dosing of both of those things. So it's interconnected, so if I separate them, I've created a challenge for myself. Right, and then finally, just the complexity of it all is a challenge for algorithms. So we have proposed this approach over a sequence of papers, which we call function networks. In this approach, we model our objective function, in this case the amount of SARS-CoV-2, as computed by a collection of functions, which are arranged in a directed acyclic graph, just like here, right? So each of these circles is a function that takes some inputs, in particular this molecular structure, and then outputs, a vector of numbers, in this case, how permeable cell membranes are to that particular molecule, right? And then we can build an AI model, a predictive model that knows about everything, that knows about all of these functions and can reason about the relationships between these functions, and that incorporates data from experiments where we pick a sub-collection of nodes in the network, and then we vary some of the inputs and look at the outputs. I'm motivating this approach in the context of drug design, but it's broadly useful. Here's an application in reinforcement learning for robotics, where you are interested in designing the trajectory of a robotic arm. You can divide that up into time steps, and the state of the robotic arm as it leaves one time step impacts the state of the arm as it goes into the next time step, and there you can create a directed acyclic graph. To explain how this method works, the methodology within this method, I'd like to use this simple example, which just has 2 nodes, where this is the decision vector that I'm trying to choose. This is the black box function, h of x. It produces some outcome y, and then there's a known function g that transforms this y into my objective function. This is kind of our first work in this area from a nice single paper back several years ago. An example of where this shows up that I'll talk very briefly about at the end is in solving inverse problems, where I'll present an example where we work with some people who do electron microscopy who are interested in calibrating their the method that they use to learn. So in electron microscopy, we shoot beams of electrons through a material and do that in order to understand what's in the material. And they have there's some key parameters in their method for doing inference that need to be equal to what's true in the real world. So it's basically the position of the stage through which electrons shine in the experiment. And they have a simulator that is H that produces, when you propose a vector X of real world parameters that are governing the shape of this stage in the electron microscope, that makes predictions for different statistics that we should observe in reality. And they're looking to solve an optimization problem where Yj is what they actually observe for that statistic, Hj of X is what their simulator predicts they should observe, and they're trying to find the X that simultaneously causes each of these predictions to be equal to the real world observation. They're trying to minimize this, in their case, sum of squared errors. Right? So this is a composition, a function composition, a function network where the black box function h of x produces an output, which is then processed through a known function. That's the function that computes the negative sum of squared errors to produce our objective. So how do we apply ideas from Bayesian optimization to this problem? So remember, in standard Bayesian optimization, we have this model for the objective function, and we have this acquisition function which quantifies how useful an experiment is towards the goal of optimization. How does this acquisition function work? So one of the acquisition function I'm showing you is called the expected improvement. So think about, this particular point x. We have an estimate for the objective function shown in the solid blue, and we have a shaded region which quantifies uncertainty. And the statistical method, what it's really doing at that position is it's giving a probability distribution for the true value of the function at that point. That probability distribution, what we do with it is we say, hey, how useful is it to perform an experiment at this x? Well, if we sample at that x, we'll get some improvement in our solution, right? So we'll observe f of x, and if it's better than the previous best solution we've seen, whose value we'll call f star, then the improvement is just that difference. But if this thing is less than this thing, so this difference is negative, then the improvement is 0. We don't know what this improvement is, but we know a probability distribution for this quantity, and so we can take the expected value of this improvement, and that's what this acquisition function is. Okay? So we can adopt that same idea in other statistical models. So just like in standard Bayesian optimization, we fit a Gaussian process to our observations, and then we find the x that maximizes this expected improvement. When we do this function networks approach, which we also call a gray box approach, we fit, instead of fitting the Gaussian process to the overall objective function, we fit it to the thing that we don't know. So each of these individual functions, in this case there's only one, this h of x, we model the relationship between inputs to that function and outputs. Okay? And then we apply the same approach for creating an acquisition function, where we take the expectation of the improvement, but now the improvement is coming from a statistical model that understands the relationships between, the nitty-gritty, this function network. Right? So we can sample from our Bayesian posterior probability distribution on h of x, push it through our known function g, and get samples from this, and then by averaging, come up with an estimate of the expected value. And then when we perform that experiment, we don't just observe the final result, we also observe the inner function. When we run an animal study with a mouse, with a candidate for Paxlovid, we observe not just the amount of SARS-CoV-2 in the mouse, but we also can probably measure the amount of nirmatrelvir and the number of copies of, we would observe a variety of other biomarkers within the mouse to get a better understanding of what's happening. So let me just show an example on a simple instance of this this function network that has only one unknown function. I have a scalar input to a simulator. H of x is the difference between the simulator's prediction and observed data, and I'm trying to solve this sum of squares minimization problem. So what standard Bayesian optimization would do is fit a Gaussian process to the objective function, which is h of x squared. Here I'm showing that when I've observed at 3 points. Okay? And then it would compute the acquisition function from that statistical model, and it would be maximized, for example, here. Okay? And I would choose to run the experiment where this vertical red line is. This alternate function networks approach sees more information, right? So it sees h of x, not just h of x squared, and you see that in the top plot on the left. Okay, so it would fit a statistical model to h of x, and already you see that something interesting. So one of the observations has a negative h of x, and two other observations that are adjacent to it have positive h of x. I'm trying to find the x where h of x is 0. So if h of x is continuous, I know that in between this middle point and each of the edge blue points, h of x must cross 0. So I already have narrowed down the space of locations for my global optimum. We take the statistical model on h of x, and that implies a statistical model on h of x squared. And under this statistical model on h of x squared, I compute the expected improvement. And it recommends measuring at a very different place from standard Bayesian optimization. In particular, it recommends measuring here, which is sort of roughly halfway between a point with negative h of x and a point with positive h of x. So it's kind of has the flavor, it's figuring out organically the idea that maybe bisection search is a good idea. I have, a point where the function is higher than my target, a point where my function is lower than my target, and then I want to measure somewhere in between. This is a test problem that solves, it's an inverse problem, and I'm showing the number of times we've evaluated the underlying simulator that we're trying to calibrate to some real world data. And I'm showing on a log scale what we call the simple regret. So that's the difference between the best solution I've found and the global optimum. Lower is better. So I'm showing a collection of classical Bayesian optimization methods. In particular, this light blue one is expected improvement. And then I'm showing methods that are based on this additional information, this function networks information. And this is the one that I just explained, expect improvement with this function networks information. And you see that it's, like this is 10 to the minus 1.5, and this is like 10 to the minus 6. So it's a 4.5 orders of magnitude improvement in terms of search efficiency. So by getting extra information, you're just accelerating by a huge amount. I've explained these ideas in the context of a simple function network, but they extend naturally to more complex ones like the Paxlovid example, or to this is a biomanufacturing kind of context in which I'm trying to make maybe some antibody, and I do that in a sequence of steps, where I grow my cells, and then I'm like collecting protein and filtering it, and in each of those stages, if I'm interested in optimizing my biomanufacturing process, I would be observing things like the amount of protein, the quality of protein, things about the health of my cells. These methods significantly accelerate performance on a variety of test problems, but I wanted to just kind of briefly show you some real-world experiments where my collaborators have used these ideas. So this is joint work with a team, actually from before we worked on these function networks methods, and then I realized retroactively that what we were doing was applying these methods there. So this is a team based at Northwestern and at UCSD, where we were interested in developing controllable protein labels that you could engineer into the genomes of model organisms to understand the impact of biochemical perturbations on their activity. Right, so you've probably heard of green fluorescent protein. This is a way to make tissues glow. The challenge with green fluorescent protein is great, and it won a Nobel Prize, but you don't have the ability to turn the label off to make it stop glowing green. So my collaborators proposed this idea of creating labels that you can turn on and off. And the way that this works is we would create a short peptide that doesn't fluoresce until a chemical reaction attaches some fluorescent thing to it called phosphopantothene that you can conjugate, a fluorescent dye to. And then the goal was to create a pair of peptides that could be labeled independently, could have this like fluorescent moiety attached to it by two different chemical reactions. So it's really about finding peptides that are substrates for chemical reactions in a specific way, basically designing for specificity. This is a picture of the experimental readout. So they have a membrane, basically a piece of paper, and to the membrane they use a peptide synthesizer to lay down peptide in a circular spot. And then they pick one of the enzymes that they're interested in using to attach a label to the peptide. They wash the enzyme over the paper in the presence of this fluorescence label. And then they take a photo, and this is what you see. You see some spots, are quite dark. So those had lots of fluorescent label attached. And then some spots you can't see anything. Right, so we could get in batches of roughly 500 measurements of enzymatic activity. This is a simple function network where I have a peptide that comes in, and I'm observing whether it's labeled by reaction 1, this is one kind of experiment, labeled by reaction 2, that's a separate experiment, and then they were interested in finding short peptides because we don't want the peptide to interfere with the biology of the organism. Right? And you could replace this by some sort of functional assay on whether it affects
protein folding or any other measure of activity. And together these jointly determine an objective function which is looking for specific peptide labels. Here's in silico results comparing our method, which is called peptide optimization with optimal learning, comparing it to a variety of other kind of benchmark methods. And so in silico, we find the probability of finding a short hit, so that's a short peptide sequence that is specific, is labeled by one reaction and not another, grows rapidly. And then we also demonstrated this experimentally over the course of 4 rounds of experiments, finding a number of peptides that were selectively labeled by these 2 different enzymatic reactions. This is the name of the enzyme, ACPS and SFP. And then this is a picture of a membrane that was used to demonstrate 2 of the lead selective peptide substrates, where they printed onto a membrane using peptide 1, which is labeled by enzyme 1 and not enzyme 2. And over here on the membrane, they printed the different peptide, which is labeled by enzyme 2 and not enzyme 1. They printed 2 copies of these, and in this experiment, they watched it with enzyme 1, and so you see all these, and then here these gray ones are printed just with a control peptide. You see that like this side is labeled by enzyme 1. This is not supposed to be labeled by enzyme 1, and indeed we see, only fate readout, and then here we do the opposite experiment where we demonstrate that it's labeled selectively by enzyme 2. And then maybe I'll close just with one last example, demonstrating these methods in not a drug discovery example, but a material science example, which is electron ptychography. So electron ptychography is a type of electron microscopy where we record more information, and that allows us to learn more about the structure of the material through which we're shining electrons. And in particular, my collaborator, David Muller at Cornell, has demonstrated that you can get sub-angstrom resolution through this technique. So maybe okay, so you have typically a crystal, and you're interested in understanding in this crystal, where are the atoms in the crystal? So it's a thin slice of crystal, which is kind of here, and we have a beam of electrons which is shining through the crystal, and then we have a detector below. And the detector is a 2-dimensional detector, and you're seeing diffraction patterns. And then you move this makes it look like you're moving the beam of electrons around, but what you actually do is you move the sample around the crystal, and you see how the diffraction pattern changes as you move the crystal. They then solve a large inverse problem using PyTorch on a GPU to figure out which arrangement of atoms allows the diffraction patterns that are observed to match the diffraction patterns that are predicted by the physics corresponding to a proposed arrangement of atoms in the material. The challenge that they have is there are some key parameters that are difficult to do gradient-based optimization over. In particular, the X tilt, the Y tilt of the stage underneath the electron beam, or actually of the sample as relative to the electron beam, and then also the thickness of the sample. So what they want to do is solve this optimization problem where So up here you see an example of what the image looks like. This is like what a reconstructed material looks like if you use the incorrect tilt, and then you get a more accurate estimate when you use the correct tilt. The way they get the tilt is they solve this optimization problem where x is 3 dimensions, the thickness, the tilt x, and the tilt y, and they're able to reconstruct what kind of an average version of the data from the electron microscope ought to look like under a proposed collection of these parameters x, and then they compare it to what they actually observe, and then they want to minimize the sum squares. This is summing over all the pixels in the image. So we view this as so previously they were using standard Bayesian optimization, which is better than kind of hand tuning the thickness and tilt, and it was also better than just doing exhaustive search over all thicknesses and tilts. But we came and said, hey, this is a function networks problem for the following reason, right? So I have a vector x, which goes through a black box function, which produces a simulated image like this. So that's y of x. And then if that simulated image goes through a known function, which calculates the sum of squares, and you're trying to minimize that, right? So let's just apply the methodology that I explained to help accelerate your electron microscopy characterization. And it works really well. So here's a plot showing one measure of performance, which is the sum of squared errors, versus the number of times you have to call the simulator, the expensive simulator. They'd like to be able to do this more quickly. This red line is using function networks, Bayesian optimization for electron microscopy, and these are some standard Bayesian optimization approaches and then a random search baseline. Another measure of quality is the kind of the error that you get when looking at recovering things like the correct tilt, the correct tilt in the 2 dimensions and the thickness, and you get substantially tighter estimates of these different sample quantities, both like less uncertainty and more consistent across across runs. Okay, so those are just 2 examples of how we've applied these approaches in the context of AI for science applications. More broadly, I would say that it's very important to integrate experimental design and optimization methods within the fact that real-world drug discovery efforts require this kind of complex function networks view. And then by doing so, we can substantially accelerate progress. So thanks very much.